Hi, everyone. So yes, I'm going to be telling you today about fast-forward fragments and specifically walking you through our XChem technologies and also highlighting some of the ASAP learnings and walking you through one of our recent ASAP exemplars for fragment progression. So just a little bit of the overview of the talk today. I will reintroduce you to how XChem approaches the design, make, test bit of the DMTA cycle for fragment progression. And as mentioned, I will walk you through an exemplar where we applied FFF together with MedChemica for lead optimization on the ASAP program. And then I will walk you through some details of the fast-forward fragments technologies, and then also finish off with just some lessons learned so far. So, just a quick comparison between high throughput screening and fragment progression on the right-hand side. So, XChem is a high throughput structural beamline, so we can leverage that that potential to generate lots of high quality structural data. And just to give you an idea of why we focus on fragment progression, and why we think we can create our own luck. High throughut screening libraries are typically very big, and they could, there's a psychology involved where you'll go through these massive libraries and get a couple hits, and everyone gets very excited about those hits, and there's a motivation to progress them. From our side, our challenge from a fragment progression point of view is we use much smaller fragments, and we typically screen much smaller libraries, in the order of 500 to 1,000 compounds, orders of magnitude less than the high throughput screening libraries. And the thing is, once we've got these structural hits, they may not give the same readouts as what you'll typically get from a high throughput screening readout. And our challenge is to take these hits and use them to design follow-up molecules that can progress them rapidly. And I think that's the key thing here is developing systematic approaches to how we approach this fragment progression problem challenge. All right, so just in terms of the design, we are a fragment screening platform, so you can think of our fragments as small Lego building blocks. And once you've got these structures, how do we combine them, to get our Death Star or our hit a compound that we can take to a lead, etc. And one of the key things that we've from our perspective, we've learned from doing on several projects is that merging seems to be the way of combining fragments to get to on-scale potency quickly. And that is very much the focus of our design efforts and progression efforts from XChem. So typical approach, of course, you get your fragments, and what usually follows is many design, make, test, analyze cycles, and before we can eventually get to our end goal. And each cycle does get smaller, but you start adding all these iterative attempts up, and it does become very expensive, from a resource which will be cost and time to execute these types of iterations. And we are a user facility, so we do try and make our technologies available to academic and industry users. And not everyone can obviously afford this drug discovery, iterating through many design, make, test cycles. So how we think about this problem is, what you got on the left-hand side here is a target with many structures bound to the target, and how do we take this rich structural information and convert it into to hits and leads? And collectively, we've come up with this design, make, test, analyze cycle that we're calling fast-forward fragments for rapid progression. And how we define that is, in the order of months, 1 to 3 months. Really very modest budget of around 20,000, and we aim to explore up to 1,000 plus compounds get 1,000 structures and 1,000 affinity data points. And really what we'd like to do is turn this cycle, this iterative cycle, reduce them to one pass. And the ambition is, can we get to that on scale potency, around the low micromolars with a single pass of this design, make, test cycle. I will not talk about the analyze bit. We specifically focus on the design, the make, and the test bits. The analyze bit is, of course, to follow, and there's obviously an exciting opportunity to use all this data that we generate for machine learning and other applications. So collectively, these are just a really high-level overview of some of the technologies we have at XChem. And starting on the top left, on the design bit, nothing's going to happen without fast data dissemination. And I'll also highlight it needs to be collaborative. We've developed Fragalysis, which is a web application for uploading structural data from XChem. It also allows the upload of in silico molecules and comparison of the in silico molecules to the experimental structures. And there's a lot of, it's ongoing effort, but, the idea here is, can we allow the structural biologists and medicinal chemists to communicate with one another through data sets that are well curated and annotated. So moving on to the make bit, the very important bit that that's central to our chemistry efforts here. So we've got a digitized chemistry platform, which I'll tell you about a little bit later. But the most important thing to note here about our approach is that we focus on tractable chemistry that we know we can execute for high throughput chemistry, and we focus on elaborations that do not include the points where we actually have to execute the chemistry. And that's really how we elaborate around the space, around these merged designs. And the test bit, like I mentioned, we're a high throughput beamline, which is automated. And the other key thing with the test bit is that we've also soak crude reaction mixtures, so that we found this to be quite effective for saving a lot of time, because obviously purification can take a lot of time, and that can be a real bottleneck. And we have several examples of crudes being used, but this is ongoing work to see how effective these crudes are when soaking them. And as mentioned, collectively the idea is this will create very large AI data sources for predicting ligand binding and drug discovery. So I'm going to get straight into an exemplar now. This was a project done with David Cousins and Ed Griffin from MedChemica. They are on our lead optimization part of the asset project, our P5 project P5. And this project, we basically got together and saw how we could leverage some of our high throughput chemistry and their MedChem smarts to help them with a lead optimization problem. And basically, this was for the dengue virus and Zika virus target, NS2B, NS3. And what they did with this initial sort of approach was they added a bunch of basic groups to one of the vectors. I can't share any of these structures with you because it is confidential. This was a way to gain reliable potency. You can see moving from the monobasic to dibasic form. But the problem with this is that it increases the polarity, and also gave issues about reducing the permeability in cells. And so David and Ed approached us and basically asked them, can we try explore a chemical space to substitute this basic group and just help with still keeping improving our inhibition, but helping reduce the polarity and increasing the cell permeability. So a little bit about the elaboration approach used by MedChemica. What you're looking at here on the top right-hand side is a building block. So you can imagine this amine is used in a amidation reaction. And when counting the distance, so these are the number of atoms from the reacting group, the idea is that you'll have different pharmacophores, And what the elaboration approach does is basically identify different pharmacophores at increasing distances away from the reacting group. That's the one feature they use. And then the other one they used, of course, was the shape, where they basically try to cluster these building blocks by their shape, and you can see this plot here with some of the distribution. And the idea was then to sample from that distribution to get different versions of the shape of these building blocks. It's also important to note that it was also based on size. So the idea is that when you can obviously get very elaborated and very big building blocks that would be expensive or complex and difficult to make. And the idea was also to constrain that to be slightly smaller, which is great for early high throughput chemistry and elaborating these building block sets. So this is what you're looking at here is the actual chemical space. So the thing to note is here, I've mentioned the distance from the reacting group that's represented here on the y-axis, and on the bottom are the different pharmacophores. I think the first thing here you'll notice, the numbers represent the number of building blocks that were actually identified for each pharmacophore and from a particular distance from the reacting group. And the first thing I'll point out, yes, there were certain pharmacophores that were not sampled, and I'll get to that in a minute. The med chemical guys certainly did look at that later, but we still decided to proceed with this space. I mean, you can see on the left is all available building blocks, and on the right, the actual building blocks that were picked for the synthesis. And you can see that we lost even more of the chemical space coverage with the selected building blocks. But like I said, we did proceed with these, and I'll show you in the next couple of slides, we were still able to get some results. So a bit about timelines. We got together on December the 11th last year, and basically by April, we had established some SAR and also validated data with pure remade compounds. So, in the space of, I'd say this was actually in the space of approximately 3 months, if you consider the December Christmas break and New Year break, etc. This was, this was a quick turnaround to get from designs to readouts. And I'll just mention Matthew Golding, our chemistry technician, was very much involved with this and helped drive the rapid turnaround of the synthesis. And of course, there's a massive effort, a collective effort from the XChem team, who also had a look at many hundreds of structural data sets for this project. So in terms of focusing a little bit on crudes and the challenges with using crudes, one of the key experiments that we did, and it's really super obvious one, is also submitting all the reagents and building blocks for in a single point measurement, and if they did show any inhibition for a further full dose response. And this was key, of course, because you can see, in this table here, I mean, it was a simple amidation reaction. These are the reagents involved, the Hatsuzi activator along with the base type here and the solvent DMF. And basically what this showed is that the very low probability that these reagents and solvents had an impact on our inhibition measurement. And likewise was the case with the building blocks, which I haven't shown. So what happened after the crudes were submitted for a single point measurements, the crudes that showed inhibition were then submitted for a full dose response. And then from that set, there was a triage that Ed and David did to basically select a bunch of compounds to ask Enamine to remake that are purified. And what you're looking at here is the dose response of the remakes against both the dengue virus and the Zika virus. And basically what you're seeing here is 3 compounds that had pan activity, and also were, in the low, focusing on the dengue virus in the low micromolar, which is where we wanted to get to after going from that monobasic to dibasic scaffold. So, objective achieved. And I think the thing to highlight, that we're really excited working with Ed and David on this was just the speed at which this happened. And I will just also mention thank you to Noah from the Weitzman, who helped turn over both the crudes and the pure analysis for hundreds of these compounds in a very rapid time. So a note on comparing crudes versus the pure compounds, and it was interesting. What you got here on the y-axis is the crude inhibition response, and on the x-axis is the pure compound response of the remakes. And what was interesting is that we had really good correlation, and that was in red, the little red dots. That was interesting because it was for a particular scaffold, which seemed to show a very good correlation. With the other scaffold, the correlation not the best. But interestingly, it could be, something we need to look at is the impact of these different scaffolds on inhibition, this correlation readout. But you're not, the correlation is, not massively far off. So the learnings from this, basically, I mentioned earlier on, this was the space that we explored. These were the elaboration space. I'll remind you, these are the pharmacophores and the distance from the reacting group. And if you recall, I mentioned there were some gaps in the available building blocks and what were selected, in gray. And what Ed and David have done is improved this building block selection. And what they've also done is made sure that when sampling the available building blocks, that at least 5 building blocks from each of these subgroups, or each distance group is selected for synthesis. And this is an iteration that we're busy working on now. We've got the building blocks and synthesis pending, but you can basically see how the space is way more comparable to what was available in the catalog, and we're covering more pharmacophores with more distances from the reacting group. All right, so I walked through the example. I just wanted to show you what what we've done recently, and what I'll just spend a few minutes on now is just think how we think about these things. And we working with MedChemica, we think of MedChemica as a potential user, and this is very helpful in how we put all these bits of the pipe together. So our approach here is always user focused. And what you're looking at here is the bits of the pipe that we think are important, and from the point of the view of the user. Starting with the data dissemination bit. I mean, many users may not have really available cloud computing resources or the computational expertise to actually help themselves get their data out, and also tools like that will give their data context with the curation and annotation of the data sets. The next bit is the design bit. We have a lot of users that are not medicinal chemists or have no idea what to do with their data. And, we also want to help them answer this question of how they effectively explore the chemical space available. On the make bit, not everyone may have synthetic chemists or accessible to or access to a chemistry lab. The other key bits here are how do they spend their budget grants on compounds. And, we also would like to encourage people to actually make the compounds themselves. And I'll explain a little bit more in detail about our digitized chemistry platform in a bit. So collectively, this is the elaboration approach that we're taking with expanding the chemical space from fragment hits. So starting on the left, you've got your fragment screen. You've got your data dissemination bit. I will just highlight these are the numbers of, you can think of these as the number of potential data sets. So how it will work is you'll get your data, your structural data, there'll be a data dissemination bit. And this will move on to where we'll identify merged pairs. Like I said, we really focus on merge designs. Identify mergers. At this point, there's a curation and selection step where the merge quality is assessed and selected. And we can either go in two directions. We can either go straight to the catalog here, or we can actually progress with this selection. And the way we progress with this, what we're calling Tier 3C, is using synthesis-directed elaborations to expand around these merge designs so we can get into the hundreds of thousands of designs. And we've got a tool called Hippo, which is the multi-objective selection tool that basically down selects the expansions to the order of thousands. And then there's another sort of curation step of looking at chemistry we can actually do on the robot, in terms of building blocks that are available, etc. And finally a final sanity check of the final pathways, chemistry pathways, which are all suggested using retrosynthetic prediction tools. And on the make bit, we get to CAR, which is our chemist-assisted robotics platform. We'll actually synthesize these targets in the order of thousands and then send them to XChem for getting structures. And just these are the folk that we see being involved. We need at least a structural biologist, a medicinal chemist, and we provide compute resource and obviously support from XChem in red. So I'm going to talk a little bit about CAR now, just an overview of CAR. So it's a digitized platform, a chemistry platform. And I think the main thing just to highlight here is that we piped a whole bunch of tools that help people get chemistry executed. And the real driver behind this is the way we encode recipes. These are digitized chemistry recipes that are also database driven, that are both human and machine readable protocols that we basically can execute on a very low cost liquid handler, Opentrons. And we're always trying to push the types of chemistry and what we really can achieve on this low cost liquid handler. So just moving on to some lessons learned about crudes, going back to ASAP. This was work done with Yasmin and Peter. And one of the key things here basically is we're working with crudes, and this involves potential solvents that carry over, or reagents and catalysts. And what happened with this particular project is a bunch of our amidation reactions basically destroyed the chickpea crystals, which you can see here in this figure here. And what Yasmin and Peter really did was really help identify over certain soaking times of 1 hour and 3 hours. This is the resolution of the crystal structure of the protein after being soaked. And you can see that under certain conditions, it's very detrimental to the resolution. And the key culprit here was the HATU activator, which is key to the amidation reaction. So this led us to work on a bunch of workup. This is chemistry geekery. This is basically use an aqueous workup to remove the HATU. So it's water wash. And the idea is to use this liquid handler to extract the organic phase. The challenge here is, in blue is the water phase with some food coloring in it. The challenge here is that the Opentrons halogenated solvents are not compatible with the pipettes. So you have to use solvents that are non-halogenated, and they're always less dense than water. So you have this challenge of where exactly your pipette tip actually needs to hit down to actually extract only the organic phase. And basically we did a bunch of calibrations of just determining how the heights of this water layer, a very simple linear regression to estimate the height, and it actually worked relatively well. We did soak these again. Unfortunately, no hits from that, but I suspect we have a bunch of mass transfer issues that we'll have to fix. Other types of workups that we look into is using solid phase extractants, and this allows us to do Suzuki reactions, for example. And you can see carefully, there's a bunch of vials that are colored a very dark color, and that's the palladium catalyst. That also works quite effectively for removing the catalyst from the reaction mixture. So just going back, we were diverted and we did a whole bunch of work on the workup to remove HATU. And part of the reason for that was that we couldn't soak any more reagents at the beamline. There was just a shutdown period. And it actually just turns out that when you do soak a bunch of other activating agents, there's DCC that we could that was actually all right with crystal quality and soaking at different concentrations. So that's another approach. So these controls and soaking controls are key to using these crude reaction mixtures. And just future work planned, quick summary. This is basically the targets on the left. And and this is working through the design make test bit. Everything in green is basically done, and in orange is pending. And I think the key ones here are the work that we'll be doing on the RDRP and the dengue and Zika viruses. We have a lot of follow-ups planned and a chemistry planned, as well as catalog compounds planned for those targets. And that's me. Thank you very much.